right, let's get started. So I hope everybody had a good fall break. I know everybody feels like it could have been longer, myself included. Next year we'll have a full week. I enjoyed some time off. I saw my family, did some fishing. It's a nice time. But I also graded exams over the weekend. So I have passed back year exams. These are the class results here, the average being about a 74%, but it was very bimodal. There are a lot of A's or a lot of F's, all right? It is a tough exam, and I know what happens around midterms when you have other exams as well, all right? But that being said, I'm gonna stick to what I did last time, all right? I did the statistics for every single question and figured out which one of those over 50% of the class got wrong. So automatically, I gave you a couple points back if you got that wrong. All right, so let's see. Short answer 3C, I gave you a point back if you got it wrong. I think that one was asking about photorespiration. Short answer 8A, I gave you a point back if you got it wrong. So those are the 2 steps of fermentation. And then short answer 9, right? We actually did pretty well. We didn't make the 50% threshold, but I realized I worded that question a little wrong. There's some errors there. I gave everybody half a point back, regardless if you got it right or not. Right? Now there's other questions that everybody kind of did poorly on. And these are the ones I have in green here. And these are the ones I'm going to give you an opportunity, if you got it wrong, to get half points back again. All right, so I have sent out an email with the instructions on how to do that. Same exact way as last time. I want you to correct it. I want you to write a ratio down. All right. That being said, I will say, I was slightly disappointed on a couple of them. Right? So a couple of these questions you saw several times in lecture, and still half the class got it wrong. Right? I debated with myself on whether or not to give you a chance to get them back, but I'm going to hold true to my word of that 50% threshold. I'm going to give you a chance. All right? But that goes to show you questions that are asked you in class can be present on the exam. All right? Particularly the Kelvin cycle. Right? You saw that diagram a couple of times. And I specifically told you what 4 molecules you had to know. I saw a lot of blanks there. Otherwise, I mean, this exam wasn't terrible. Right? The average is still 74%. Right? That's still a really good average for a science class, especially a science class like Bio 101, where we have to go through materials so fast. Right? It's a tough class. Right? And unfortunately, Bio 101 really sticks to traditional ways of measuring your success in form of exams. But remember, there are those scientific literacy assignments, your content checks, and your lab grades that all play into your midterm grades. All right? Any questions on this past exam? You have a question? Sounds good. So we're going to move right along into the next bit of content. All right, so this next unit, we are really going to focus on genetic material, how it replicates, how a cell divides, as well as a little bit of genetics. All right, and we're going to jump in by just doing a lot of review today. I know we had some of this material previously, but I wanted to do a good chunk of review to catch everybody up because you haven't had this material since, second week of classes here. All right, so we're talking about genetic material, right? And so that would be like DNA or RNA. And in order to be genetic material, there has to be 4 criteria that need to be met. That 4 criteria is that genetic material has to have information in it, right? So for example, DNA has information on how to build proteins and information on genes. It has to be able to replicate. Okay, we'll talk about that on Friday. So DNA is able to replicate itself. It has to be go through transmission, which means it has to be able to be passed from parent to offspring. And finally, there has to be variation. Right? There's variation in our organisms that we see every day. That variation has to be accounted for by the genetic material. And we see variation in DNA. Right? My DNA is very similar, but also different from your DNA. So there is some variation. Right? All living organisms are going to use DNA as their genetic material. Right? Pretty much any organism you can think of is going to use DNA. Right? Now, viruses are the oddball here. Right? Viruses are actually considered non-living because they're not made of cells. They can't reproduce in the same way. They don't contain DNA, but they still contain genetic material. They contain RNA. So I just want to throw that little caveat out there. All right. So all these organisms use genetic material, mostly DNA. RNA is still going to be present and it's used by our cells in many different ways. And so we want to go over what makes up DNA, what makes up RNA. And like I said, some of this is reviewed. So DNA is deoxyribonucleic acid, right? This is double-stranded. It has that classic double helix shape to it. It's going to stay in the nucleus of the cell. It's not going to leave the nucleus of the cell. And it's going to store all of our genetic information. Versus RNA, or ribonucleic acid, it's only single-stranded. It can leave the nucleus. And RNA contains kind of the instructions in order for our cell to build proteins. Now there's more differences than just these, and we will see these on the next few slides here. But in order to understand DNA and eventually DNA replication and transcription and translation, we kind of have to understand the anatomy of our genetic material. And so we're going to go over that in this lecture today. We're going to start at the very, very basic building blocks of DNA and RNA, which would be our nucleotides. Right? They should be familiar to you here. Those nucleotides, they will combine together and form a strand. Right? RNA having one strand, DNA having two strands. DNA, with those two strands, will form that double helix. Once we have our DNA, they can condense into chromosomes. Chromosomes contain not only the DNA, but also different proteins associated with condensing that DNA down. And then finally, we have the genome. Right? The genome is the entire complement of all of our genetic information in ourselves, in other organisms. So like I said, we're going to start on nucleotides. This should be a review, good slide back into lecture after fall break here. So nucleotides, we know they're comprised of these three components. Right? There's a phosphate group, there is a sugar, it's a pentose sugar, which means it has 5 sides to it, or 5 carbons to it, and it has a nitrogenous base. All right? The difference in DNA versus RNA is the type of sugar that it has. So DNA has this deoxyribose versus RNA has this ribose. The only difference between them is the addition of an oxygen off of this 2 carbon here. So ribose has an oxygen, deoxyribose does not. Right? But that does make them a different sugar. Very similar, but different sugar. And then we have our nitrogenous bases, right? With thymine, do we have thymine up here? Yes. Thymine being only in DNA, and uracil only being in RNA. Okay? And we're going to look at those nitrogenous bases in a little bit more detail, and this is new information. Right? Because we can take these nitrogenous bases and break them down into 2 categories, which are quite important for how DNA builds its double helix. So we have pyrimidines and we have purines. Pyrimidines, they have a single ring structure to them. So that would be the cytosine, uracil and RNA, and thymine and DNA. Right? Just a single ring to them. Versus the purines, they have a double ring structure. So this would be our guanine and our adenine here. Now, purines are always going to pair with a pyrimidine. Right? So we're always going to have guanine pairing with cytosine, adenine pairing with thymine and DNA, and uracil and RNA. Right? So purines and pyrimidines always pair together. So this is quite important information to know. This is information, these details I might ask you on an exam. And so it's good to have ways to remember which is which. There's a lot of mnemonic devices out there that can help you out. I put one in the notes of this slide, but one of them is cut the pie. So if you say cut, which is C-U-T, cytosine, uracil, thymine, right? Cut the pie, which is P-Y of pyrimidines. That's one way you can remember this. Right? Another way might be pyrimidines is a long word, and it's associated with a smaller molecule, versus purines is a short word associated with a larger molecule. So kind of opposites there. Right? Whichever way makes sense in your mind to remember these, great. If you did a quick Google search, I'm sure you would find many other ways to remember these as well. All right, so if we put our nucleotides together, we are going to get a strand of genetic material. Right? So remember those nucleotides, those are the monomers, those are the building blocks. We put them together and we get a polymer, and those polymers are nucleic acids. Sometimes they are written out instead of a nucleic acid, we'll call them a polynucleotide. So polynucleotide. Right? So these nucleotides, they are covalently bonded together. And this covalent bond is specifically called a phosphodiester bond. It is a bond between the phosphate group of a nucleotide and the two sugars on either side. Right? So a phosphate group coming off of the sugar from one nucleic acid being bonded to the sugar of another nucleic acid. This is going to form a backbone, in which we specifically call this a sugar phosphate backbone. Right? So our sugars and our phosphate are all bonded together. You can see it's highlighted in red here. This is our backbone. Right? And those nucleic acids, right, or those nucleotides, I should say, they are going to project away from the backbone, and they'll all be on the same side. All right, I know you've seen this image before, but I just wanted to emphasize that it is a phosphodiester bond, and it's happening in this type of reaction that we've seen before. It's a dehydration reaction. So in our phosphate group, we have this OH group. We also have this hydrogen off of our third carbon here. Right? These are going to combine together to create water, which will leave the reaction, and what's left behind is our phosphodiester bond. All right, so nice simple dehydration reaction going on here. But if we talk about a strand of nucleotides, right, if we talk about a polynucleotide, we have to talk about its directionality. Right? So these strands, they have directionality to them, and this becomes particularly important when we talk about DNA replication. So if we look at our sugars on our sugar phosphate backbone here, right, we can label the carbons in our sugar. Right? Starting on the right, our first carbon will be 1, second carbon 2, 3, 4, and finally our last carbon that kind of projects off of that fourth carbon is the fifth one. Right? We often call this like a 1' carbon or 5' carbon. Right? So when you write out a DNA strand, you would write it out in the direction going from the 5' to the 3' carbon. Right? So starting at this 5' carbon that kind of juts out here, all the way down to the 3' carbon on the last nucleotide of the strand. right? So if I were to write out a strand of DNA, I would start by saying I'm going to show you the directionality, so I'm going to write down the 5' first, and starting with my 5' carbon. Now I'll have thymine, adenosine, cytosine, guanine, and that's going to end at my 3' carbon, right? I know this can be kind of hard to understand molecule. All right, so let's look at these questions. So if we're thinking about DNA, right, and RNA for this matter, what is the monomer called? It's a nucleotide, right? It's that molecule with the phosphate, the sugar, and the nitrogenous base. What about the name of the polymer? The whispers, yes. Nucleic acid, yes. We could also call it a polynucleotide, but the formal name would be a nucleic acid. What are some ways that RNA and DNA are different? There are multiple things here. Yeah, the sugars are different. So in DNA, it's missing oxygen. It's deoxyribose versus ribose in RNA. What's another difference? Yeah, DNA has 2 strands and a double helix, and RNA has a single strand. And what might be the last difference? Yeah, RNA can leave the nucleus, and DNA will stay in the nucleus. Yeah. Now there's some other differences in there. We could say another difference would be RNA has uracil instead of thymine as a nitrogenous base. And then how do we tell directionality on DNA? I heard it. It's the carbons, right? We label those carbons, we number those carbons, and we follow where those bonds are on each numbered carbon. Any questions or things I can clarify? Mm-hmm. Yeah. It's not based on the diagram. Okay. That's why you are confused. Would it start at the carbon? It wouldn't just be a carbon to carbon, it would be all carbon. yes, if I'm understanding what you're asking here. Let me go back to that slide. So if I were to write out this diagram as a directionality, right, I go 5 prime, I see which end is my 5 prime, which I can see. The 5 prime end is on this side here, so it's the one left standing. It doesn't have a bond to another nucleic acid or nucleotide here. So I would go adenine, guanine, cytosine, thymine, and then my 3 prime at the very end. Yes, yes, that last carbon that's not involved in a phosphoryl. Yeah, if you write out an entire DNA strand, this can be a very long DNA strand. But no matter how many nucleotides you have in the middle, you're always going to have 5 prime on one end, 3 prime on the other. It could be 4, it could be 10, it could be 100, it could be 1,000. Yes. Yeah, so the letters are going to represent, so T for thymine, A would be adenine, C, thymine, G, guanine. Now this is not this, which is, I understand confusing now, and I'm going to make a note for when I teach this next time. Two like those together. Yeah. You can go the other way. Typically, we're going to write out the 5 to 3 strand, and then you can, if you have the 5 to 3 strand, we would know what the 3 to 5 strand is. We'll get into the next few slides here, because we know what would pair with thymine, we know what would pair with adenine, so on and so forth. So most people write the 5 to 3. But depends on the context that you're writing down this. I understand there's a little bit of confusion here. Let's go on to the next few slides. We're actually going to start talking about the strands and putting them together, and this might make things click. And if not, we are going to go over this in more detail when we talk about DNA replication on Friday, because this is when directionality plays a huge role. Let me just move my slides. There we go. All right, so far we've really only looked at one strand, but we know DNA has 2 strands. All right, and we know DNA is in this double helix. So one strand is a single molecule. Right? So in DNA, right, we put 2 molecules together, or 2 strands together, right? They're bonded together, which then makes one huge molecule with 2 strands on it. So each strand is a molecule when they're separate. When they're bonded together, then they're part of the same molecule, right? So DNA has 2 molecules bonded together, right? One strand and another strand. Now, these are huge molecules, right? Very, very large molecules. And they will be bonded together, and once they're bonded together, it becomes one singular molecule. Right? Now, the bonds that hold these strands together are going to be hydrogen bonds. So if we remember back to bonds, right, you know, covalent bonds are very strong bonds. Ionic bonds are kind of in the middle, and hydrogen bonds, they're strong, but they're the weakest of the 3. Right? And so it's our hydrogen bonds holding these together. And you might wonder, well, why are we using the weakest of the bonds to hold together DNA? It's such an important molecule. Well, we need to be able to replicate our DNA, which is eventually going to have to split the strands apart, and hydrogen bonds tend to be easier to split apart. Right? So we're always going to pair up our bases in the same exact manner, right? This is called base pairing. Adenine is always going to go to thymine. Right? And because of their structures, that's always going to have 2 hydrogen bonds. Right? And you can see adenine to thymine here. We have 2 hydrogen bonds, those little dotted lines in between our 2 molecules here. And then guanine is always going to go to cytosine. And again, because of their structure, they actually have 3 hydrogen bonds. And we can see that here with our 3 hydrogen bonds. Right? So this would be base pairing, linking up a guanine to a cytosine. or an adenine to a thymine. We can say altogether, the DNA molecule as a whole, right, when the two strands come together, that would be complementary pairing, right? So one of my strands is complementary to the opposite strand, right? So it has all of these nucleotides that complement the nucleotides on the opposite DNA strand. All right, so we've learned a lot about DNA so far, right? We know that it's double-stranded. We know that the strands are anti-parallel, so they have that directionality, and one goes 5 to 3 prime, the other goes 3 to 5 to, one goes 5 to 3, I forgot what I said first, and the other goes 3 to 5, right? I just stumbled up my words quite a bit there, right? But they are anti-parallel to each other. They go in opposite directions. They have a sugar phosphate bone, a backbone. Right? The bases will be on the inside. They are going to be held together and stabilized by hydrogen bonding between our base pairs. Right? And very specific base pairing. So thymine, always going to go with adenine. Guanine, always going to go with cytosine. I'm my words. I think I need to stop and take a drink of water here. And it's going to be a right-handed helix, which we have not talked about yet. Right? But basically, if you look at the helix, right, the strand, it's going to wind, so it's always going to wind to the right, essentially. This is something that I find very hard to visualize and wrap my head around. I always have to use an image because if I don't, I will confuse myself when I try to explain this. But if you look how it's wrapping, right, it's going to continue wrapping to the right versus this left-handed, it's always going to wrap to the left. There are instances where we have left-handed DNA. We're not going to talk about it though, right? Most DNA is going to be right-handed. Right? Does that make sense? It's a feature of DNA, tends to be an important feature of DNA. I'm not going to emphasize it too much in this class, but I want you to know that it exists. All right? As we wrap DNA, or as DNA has this double helix here, the way that the base pairs go together is going to give us this double helix. And this double helix are going to have two different types of grooves to them. Right? So just based on structurally how they combine together, we end up getting a major groove, which is this larger groove here, and a minor groove. Right? Now if you get deeper into DNA and the proteins class, you'll learn more about this. But these grooves are where different proteins will bind to DNA for different functions. A lot of it has to do with DNA replication. A major groove is where a lot of proteins will bind just because it's bigger. The smaller groove, some proteins will bind there, not as many. I did put a link in, I didn't put it on your slides. I will add it to your slides. There's a link of a video that kind of shows in a three-dimensional model the major and the minor grooves a little bit better here. I will make sure to add that. All right, so kind of some review. I know the timer already started here, but answering some questions about base pairing, complementary pairing, and figuring out the sequence that would complement that main string pair. So give it a couple minutes, ask me questions of things you don't understand, and we'll review. Base pairing is between two pairs. Very, very long. Yeah. All right, so what is base pairing? It's between the two bases, right? It's where adenine will hydrogen bond to thymine, right? And guanine will hydrogen bond to cytosine. Right? We're talking about the specific bonding between two nitrogen bases. And then what is complementary pairing? The same thing. Yeah, it's the two strands of DNA. They're anti-parallel to each other, but they complement each other. Right? So we still have base pairing and complementary pairing. We're just talking about the entirety of the two strands rather than two nucleotides. And so the sequence of that opposite strand, I'm not going to make anybody say it out loud because it's a lot of letters here, right? But it would be exactly the opposite using our base pairing rules. Right? So if we have an A, we know that complementary strand is going to be a T, right? Or an adenine to a thymine, and so on and so forth. Right, now I could write directionality on this, right, which is not on this image here, but I could say my main strand here would be 5' to 3', which would mean this complementary strand would be 3' to 5'. All right. Any questions? All right. We'll see how far we get on the last few slides here. I think we'll get too far. But DNA is a huge molecule. Right? If we stretched out a single DNA molecule, it would be about 3 feet or 1 meter long. Right? That's huge. And we have to fit that molecule, that long molecule, into a cell that's about 10 to 100 microns. Right? So a micron, if you think about a centimeter made up of 10 millimeters, and each millimeter, that would be 1,000 microns. Right? So 10 to 100 microns is such a tiny, tiny space that we have to fit our DNA molecule. And not only do we have to fit it in a cell, but we have to fit it in the nucleus of a cell. So even smaller, about 4 microns. Right? So in order to do this, we have to coil that DNA. All right, so we would say that DNA is supercoiled into chromosomes. Right, so chromosomes being a discrete unit of genetic material. Right? It's all of that DNA supercoiled around each other. So if I'm talking about supercoiled, I don't know if any of you have taken like a string or maybe even a strand of your hair when you're bored and you just start twirling and twirling and twirling, right? This would be the same concept here. We are supercoiling this string, and so it's going to wrap around itself so much that it's going to start coiling together and condensing down, right? And so when we condense it down, we are making this chromosome, right? But it's not just DNA, right? It's not just our double helix. It's also these proteins that DNA will wrap around, right? So if we talk about DNA and the proteins it's supercoiled around, we would say that this is the chromatin, right? This is the DNA protein complex. Right, so there's really 3 levels of DNA compaction that we're going to talk about. The first level is just being a DNA wrapper. Right, we're wrapping DNA around these proteins. Right, and so if we look at this image, we have a bunch of separate subunits of this protein. Right, these are histones. Right, there's histones, they're proteins, they're combined together. DNA will wrap around these histones. Right, and when we have histones and DNA, it becomes a nucleosome. Right, so combined together, this is a nucleosome. A nucleosome consists of DNA, or that double helix, wrapped around different histones. Okay? So wrapping this DNA around these protein complexes will shorten our DNA molecule by about 7-fold, or 7 times. Okay? But that's still not enough. We have to condense it even more. And this is where we get this next layer of condensing, which is called a 30 nanometer fiber. Right? It's called a 30 nanometer fiber because the fiber that results from this wrapping is 30 nanometers in width. Right? And what this is basically a bunch of these nucleosomes, right? They're put together, but they're put together in kind of like a zipper pattern or a zigzag pattern, right? Because they're offset, right? Instead of being completely equal to each other, they're offset in a zigzag, so they can condense even more together, right? And this is called that 30 nanometer fiber, right? It's just many of these nucleosomes put together in this zigzag fashion. Now the next layer of condensing, the last one we'll talk about, are these radial loop domains. Right? And so this is an interaction between those 30 nanometer fibers, or those like zipper-like fibers, and different proteins that are in our nucleus. All right. And so if we look at this image, it can be a little deceiving because suddenly we're changing how we're looking at this image. This red line is no longer the double helix, but this is the 30 nanometer fiber. Right. So this is that zipper with our nucleosomes in that zigzag fashion. Okay? And then we have these proteins that will kind of anchor them in place close to each other, and we create these loops. Right? So essentially, we're creating so many loops, kind of like what I did when this string was really, really coiled. It'll loop in on itself and condense it even further until we get a chromosome. Right? I think this is the hardest layer of folding to understand. Right? But just know that we're condensing it around histones to form a nucleosome. Those nucleosomes are condensing by being put in the zigzag pattern. And then these 30 nanometer fibers, or that zigzag pattern, is condensing even more by creating loops upon loops upon loops of itself. Right? So we're condensing our DNA. Right? And our DNA is always going to be in this condensed form. It's never going to be uncondensed. That would be a year long. It would never work. So we're always going to have condensed DNA. But DNA can be condensed to different levels. Right? Especially when we start talking about cell division, we'll see even more, DNA is going to be condensed even more. So DNA is always going to be condensed, and we could call that euchromatin. Right? It's condensed, but it's not super compact. When a cell wants to divide, it's going to become super condensed and become heterochromatin. Right? And this is when we start to see that typical chromosome shape that we are used to. Right? This really happens during cell division. These terms will come into play when we talk about mitosis and meiosis again. Right? There is some terminology associated with chromosomes that I would be remiss not to tell you about now, even though we will review this in the future. But if we talk about chromosomes, right, typically we have 2 chromosomes that carry the same genetic information. Right? So think of it as a chromosome from your mom and a chromosome from your dad. Right? They carry the same type of genetic information. So maybe they're both carrying the genetic information for eye color. One might code for green eyes, one might code for blue eyes, right? But the same type of genetic information. These would be homologous pairs. They're not identical, but they carry the same type of information. Right? When they replicate, right, they would still be homologous to each other, but either side of our chromosome, notice now instead of one yellow strand, we have two yellow strands connected together. These two sides would be identical, right? And we would call them sister chromatids. So one side is a chromatid, the other side is a chromatid. Because they're identical, they are sister chromatids. But they are homologous to the other chromosome that carries the same genetic information. And they're connected together, those sister chromatids, by a centromere. All right. I have one last slide that I'm going to go through here, because we're at the very end of talking about the anatomy of our genetic information, and that would be the genome, right? The genome, we don't just have one chromosome, right? Humans have 23 chromosomes, and when we talk about the genome, we're talking about all of our DNA, all of our genetic information, right? So we will be talking about all 23 of our chromosomes, and that would be our genome. Some organisms are going to have more than 23. Some organisms will have less than 23 chromosomes. But all the chromosomes together would be our genome. All right, I'm going to skip over this group review. We might revisit it at the beginning of the next lecture here. I just want to say, homework for Friday, there's no formal homework, but we are going to start off class with an activity. That would be best if you reviewed these slides so you know what we're talking about. And then upcoming, you do have a content check due October 30th, which is still a little ways away here. That content check is up on Moodle.